Well, a warm welcome to this talk. Now, I've got some exclusive data to share with you tonight. It's from the famous Canadian virologist, Dr David Speicher, and he's given us this data. Now, he's shown it at a conference before, but it's not yet published. Why is it not yet published? I've had a chance to look at this data. It is excellent, and the methodology to me seems beyond reproach, as far as I can tell. But it's been rejected by 4 leading scientific journals so far, and the editors of those 4 journals didn't even bother to send it for peer review. Let's hope we're not dealing with an international program of censorship. But let's get straight onto the data now, because it's quite profound and it's related to the DNA contamination. Now, here we see one of his graphs here. Now, what we've got here, these are vials of vaccine that Dr Speicher's been sent. These ones he was sent from Ireland, these ones are Canadian, and these ones are Australian vials. Now, this line here is the allowed amount of DNA contamination. Now, we can argue about this, in fact we will be arguing about this, that this allowed amount of DNA contamination is set way too high, but it is still the legal maximum amount. So is the law being, or is this legal amount being respected? Well, clearly not. So if we take, for example, let's take these, for example, these here, these JN1, this is the very latest thing when the manufacturers are supposed to have got their act together. And we find that in the very latest vials, the very latest product, we've got very high levels of spike protein. Very high levels of original sequence in blue, and very high levels of, in green here, SV40. Now, this SV40, this is actually the SV40 is a gene sequence that comes from the E. coli bacteria. It comes in the plasmid of the E. coli bacteria. These are the same bacteria that you find in your gut, in your colon, that are present in stools and feces. So the DNA from them, the SV40 sequence in that, is so important because that actually inhibits one of our own genes called P53. Now, P53 is important in humans because it's what you call a tumor or an oncosuppressor gene. It stops us from getting cancers. So if this contaminated, as we see here, SV40 promoter, and we see in the Australian vials, look, there's a positively huge amount in this, the Australian vial, the amount of contamination in this Australian sample here. So this SV40 will inhibit the activity of our P53 tumour suppressor genes. And if you're not suppressing tumours are more likely to occur. If you could somehow wave a magic wand and take all the P53 genes and P53 proteins out of my body, there'd be nothing to correct the mistakes in my DNA, and I'd probably get cancer fairly quickly, which of course I don't want. So that is quite a problem. And just look at the scale of the contamination there, the amount of spike protein DNA. So this is the DNA to make the spike protein. The DNA is present in all of these at way high levels than we're allowed, and yet these are still being injected into human beings. Is there something I'm not quite getting here? That was the first graph that David showed us. Now, just to share a bit more of the scientific data that David shared with us on the video call, and I will be playing you the whole call later. The data he collected was from PCR studies. So he did the polymerase chain reaction on these vials, and he used a cycle threshold of 12. Now, it seems that people were quite happy to accept diagnoses of COVID with cycle thresholds way over 30. So if they're happy to accept cycle thresholds of way over 30 for diagnosis, why aren't they accepting cycle thresholds of 12, which of course is exponentially orders of magnitude less than a cycle threshold of 30, where it more or less doubles every time you increase it from 12 to 13 to 14 to 15. Absolutely incredible. Now, he also told us that the SV40 DNA, not only does it inhibit the P53 oncosuppressor gene, taking away the suppression of cancer in human cells, but it also enhances the integration of DNA fragments into the human genome. Now, I didn't know this. So he's telling us here that SV40, this DNA contamination, makes it more likely that the DNA gets put into the human DNA via a process of reverse transcriptase. And of course, if it is the case that this genetic information from the bacteria to make SV40, if that actually gets into the human genome, then the human genome can make SV40 protein, which will inhibit our p53 suppressor genes. To me, that's going to make cancers way more likely, and potentially so for decades into the future, which is why this is such a concern. Now, he also told us that the threshold levels, those red dotted lines that we saw, those threshold levels they were put there for naked DNA, assuming that the DNA was naked. So if you imagine that this pen is this pen here is the DNA, then the thresholds were put in place for the DNA, which it's actually quite hard for this DNA to get into the cells. But now, in these modern preparations, the DNA is not naked. It's in a lipid nanoparticle, which is my hand. And this lipid nanoparticle finds it very easy to absorb into the cell membranes. So the DNA wrapped in a lipid nanoparticle will go into human cells 100 times more readily. This is the figure that we were given, 100 times more likely to get in. So really, these figures that we've got for the maximum allowed amount of DNA contamination should be reduced by at least 100, because the DNA is now no longer naked, finding it difficult to get into the cell membrane, but the DNA is in a phospholipid nanoparticle, or a lipid nanoparticle anyway, which means it's easy to get into the phospholipid cell membrane, increasing transfection rates by a factor of 100. So I thought that was quite interesting science that we were supplied with. And again, I'll show you the whole recording shortly. Now, here's another batch of vaccines that Dr. Speicher analysed. As we see, these ones are Pfizer and these ones are Moderna. Now, this is the historic level of contamination of DNA allowed. This was the amount allowed up to 1996. And we see that all of the contamination by the historic tolerances have been exceeded. These are the current FDA guidelines here. Now, we also see that this scale is logarithmic, 10, 100, 1,000, 10,000. So we see that these 3 marks here are actually quite significantly, with the Pfizer, quite significantly above the current levels allowed for SV40 contamination, DNA contamination. Now, the other thing we noticed about the levels of contamination that were or are allowed is that they seem to be going up. More contamination seems to be allowed. Let me just give you some of the figures on that. So FDA WHO guidelines are getting lenient. Now, in 1986, and I think up to 1996, the allowed amount of DNA contamination was 10 picograms per dose. 10 picograms per dose. Now, a picogram is 1,000th of a nanogram. So, in other words, the amount of DNA permitted was increased by 3 orders of magnitude. It was increased from 10 picograms up to 10 nanograms. And 10 nanograms, of course, is 10,000 picograms. So the allowed amount of DNA contamination in 1996 was increased by a factor of 1,000. And David said this: it's based on speculative calculations for naked DNA. And of course, remember, these guidelines are for naked DNA, not the DNA that is in the lipid nanoparticles. And we've noticed that the DNA in the lipid nanoparticles can transfect cells 100 times more efficiently than the naked DNA that these guidelines were designed for. And for some strange reasons that I certainly don't understand, he also taught us that in 2003, plasma DNA-based vaccines were given privileged status. So for some reason, the amounts of contamination didn't seem to count quite as much. So all quite strange, really. And then he showed us this graph as well here. So this is a graph of DNA contamination here. Maybe if we zoom into this one a bit, we can see the levels there, the Irish vials and the Canadian vials and the permitted amount. But then if we look at this graph actually here, this is the same graph but in logarithmic terms. So remember there, that's 10,000, that's 100,000. So this is massively over the permitted line here. These would be the historic amounts, 1986, I think, up until 1996, of 10 picograms per dose, which means that all these levels of contamination are way, way higher. And of course, remember that these levels of contamination were designed for naked DNA before the lipid nanoparticle bound DNA and its increased potential for transfection were fully developed. It's just an incredible difference. 1,000 times more contamination is now allowed. But even allowing for that, when you can see here that figure there is 10,000, that's 100,000. So we can see that that's about 90,000. If this was on a linear scale, it'd be something sticking up like a skyscraper. So really quite concerning overall. Firstly, that so much is still being detected in the latest vials of vaccine that are being produced. The manufacturers don't seem to have got their act together to clean up the amount of DNA from the E. coli contamination in their vaccines. And by now, it would be surprising if they didn't know about it. And yet the latest vaccines, it's still there, because we see here that the latest vaccines that Dr. David Speicher tested with the JN ones, and we still see that that's got very high levels of contamination. And yet we are still injecting this into human beings, which confuses me somewhat. Now, Dr. Speicher closed off with telling us about an Injection of Truth conference, and I'm putting the link to that there. There'll be video available from that pretty soon. And he also told us about the David Declaration, which I'll put the link to as well. The David Declaration embodies the many voices of the cancelled scientists and physicians who've stood up against government mandates since the start of the pandemic. Now, David is included in this because he's producing high quality scientific work. It's been turned down already by 4 major scientific journals without sending his work for peer review first. The editors have rejected his work, apparently a priori. Why didn't the editors send it for peer review? Or were these 4 editors so brilliant they didn't need to do that? Let's just go on with what this says here. David Speicher saying, "I am honoured to be part of this important and courageous declaration that calls for a return to evidence-based medicine, return to evidence-based medical science, a moratorium on mRNA technology, and recognition and support for the vaccine injured. It is my desire that this declaration stands as a beacon for Australia and the world," because it originated in Australia, "providing a framework to hold governments to account." Wouldn't that be nice if governments were held to account? That would be nice, wouldn't it? And allowing humanity to heal. As scientists and physicians, we endeavour to restore integrity to the medical and scientific fields and freedom to the people that we swore to serve. That's motherhood and apple pie, isn't it? I don't think there's much to disagree with there, really. And he also shared this. This is from the Northern Group, letter of concern from the Northern Group. And again, I'll put the link for this. And here we read, "Notice of extreme concern about COVID-19 modified mRNA vaccine safety and quality to prime ministers and governing bodies in the Nordic and Baltic countries and the United Kingdom. Excessive levels of residual DNA identified in Australian samples, and as we've seen in other samples, confirming data from France, Germany, Canada. We've seen Canada and Irish samples as well. Confirming data from France, Germany, Canada, and the USA. The introduction of foreign DNA into cells via lipid nanoparticles may damage human DNA, leading to genomic instability, cancer, and extremely serious conditions, unless, of course, proved otherwise. If the governing bodies and the governments prove otherwise, show us the data, that data is reviewed by our scientists and agreed to be genuine, then of course we will retract these concerns. Until then, that is what we are at. Thank you, David, so much for that. And I'm now going to play the full interview with David for those of you that want any details that I have left out. But from me, thank you for watching, and over to David Speicher, virologist of renown now. Welcome to this talk, and welcome back to Dr. David Speicher from Alberta, Canada. Dr. Speicher, thank you for coming back on.
Thank you, John.
Now, quite a few of you will remember Dr. Speicher from before. He's a renowned, indeed pioneering virologist, and he's been doing more completely original work on DNA contamination in mRNA vaccines. And I want to jump straight in, David, if we can, please, with this graph here. These are vials of messenger RNA COVID vaccine that have been sent to you from Ireland, Canada, and Australia. And what did you find?
So, earlier in 2023,
yep,
I looked at the vials from Canada and found all sorts of DNA contamination and SV40 promoter and enhancer sequences in the Pfizer vials.
So this is sequences of SV40 that you found in the vials.
Sequences of SV40 found in the vials. All of this work shown here is all PCR based. And in 24, I was sent 3 Australian vials, 2 Pfizer and 1 Moderna. And the Pfizer's in Australia are the highest I've ever seen to date, with a PCR-CT of about 10. So
remarkably high, isn't it?
Which is remarkably high. Late last fall, November, I was sent 6 vials from a doctor in Ireland, including the previous XBB 1.5 booster and the brand new JN ones. Now, most people would assume Pfizer has cleaned up this. They've been told by Health Canada that it needs to be cleaned up. And clearly in the brand new boosters, there is still high levels of DNA and as well SV40 sequences over the legal limit.
And the legal limit is this dotted red line that we can see.
Yes, it is 10 nanograms per dose.
And we're going to come on to talk about the somewhat arbitrary nature of that limit. But for now, the red lines are the spike protein, the blue lines are the origin of replication. So that's DNA in the plasmid.
Yes.
And the green actually, which is really, really concerning, is the SV40 promoter enhancer. And I think we should just tell people, check my understanding on this, David, if you don't mind. The SV40 sequences of DNA were first identified in a simian virus, so hence SV40. Yep. And what they will do is they will actively inhibit the expression of a gene in our own cells called P53.
Yes.
And P53 is what we call a tumor suppressor gene, which makes a tumor suppressor protein, which does just that, suppresses tumors. So if you somehow took away all the P53 genes and protein out of my body, I would presumably get cancer pretty quickly.
Yeah. Yep. And the SV40 enhancer is used to integrate DNA fragments into the human genome in cells.
So it acts as a reverse transcriptase.
Yes.
Wow, I didn't know that. Wow. Right.
And therefore, such high levels is quite problematic.
Indeed. So the SV40, it's suppressing this wonderful tumor suppressor gene, but it's also making it more likely that this DNA will be reverse. So the DNA pattern that is in the contamination could end up in the human genome is the risk.
Yeah.
Yep. Well, that is pretty bad news altogether. And just to clarify, the JN1 are the latest vials. This is the JN1 is the state of the art. It should be absolutely squeaky clean.
Yeah.
Is there any possibility that is there any way that this DNA could have got there, for example, if these samples have been preserved for too long or have been allowed to warm up? I mean, is there any other possible way that this contamination could have got there other than as a result of the manufacturing process?
Nope. All of our negative controls for all negative, and it's a PCR CT of about 12, so it's very hot. Right.
So, what does CT stand for, David?
Cycle through threshold.
Of course, yeah. Of course, so, for so-called diagnosing COVID, they were running sometimes cycle thresholds. Well up into the late 20s, weren't they?
Well, most physicians were concerned of someone with a cycle between 30 and 35, and
it doubles every threshold, doesn't it?
Yes, but if you're concerned about a PCR positive sample at cycle 35, and you're okay with a vaccine vial at a cycle of 12.
Yeah.
That's a serious issue.
Yeah, so 13 would be doubling the sensitivity of 12, and 14 would be doubling the sensitivity of 13.
It doubles. Well, it's basically a doubling each cycle, or it's a tenfold increase every 3.3 cycles.
Okay, right.
So 17 to 21 is a tenfold.
So it's quite an exponential level of increase in sensitivity.
Yes.
Yeah. Until you could be diagnosing something which is probably not there.
Yep, and therefore, based on this work, the Pfizer system is a plug and play system. It's around plasmid. The spike part each time gets changed based on which variant they want, and they've not cleaned things up at all, and it's just as bad as the rest. Now, if you compare Spike or Rory and the SV40, the levels do vary a bit, and it's because of DNA's digestion of the plasmid. In some parts, it works better than others, and it's all based on how's it being shielded and linked up and stuff. So
the plasmid DNA that's introduced into the human cell can be partly broken down by the cellular defenses.
No, it's all in the DNA's digestion part in the NUU fact.
Got it.
And so at first, plasmid was made. It's a synthetic plasmid. It's grown up in a E. coli host. It is then theorized off of that mRNA is made. It's then digested with DNase 1, and it should have been all removed, and clearly it isn't. Hence, it's a contaminant. And all parts are there. So,
so the DNA is actually coming from the plasmid that comes from the E. coli bacteria. That's the original source of the DNA.
Yeah.
And that should be digested out by DNase, but it's obviously, your graph proves that
Exactly.
didn't work in these Irish, Canadian, and Australian vials. Excellent.
Yeah. And so all of the guidelines are for naked DNA and not DNA in lipid nanoparticles. And so if you put DNA and mRNA in a lipid nanoparticle, it'll transfect cells 100 times more efficiently than naked DNA. And
really, these guidelines should be 100 less than 100 times less than they are, 2 orders of magnitude less.
Yes. Yeah. And so in 1986, the guidelines were 10 picograms per dose, and that was increased in 1996, but based on speculative calculations to 10 nanograms per dose. And
but again, this is for naked DNA.
For naked DNA. And in 2003, the WHO, they gave plasma DNA-based vaccine special a privileged status to allow a more lenient.
But of course, in 2003, there weren't any DNA-based vaccines.
There were some.
Were there?
DNA-based, so.
Right. So they gave them some exemptions.
Yeah.
Yeah.
And so based on that graph, if it's our vials tested from Canada, both Pfizer and Moderna, and based on current guidelines, some of the vials are over, and most aren't, except but based on historical guidelines in 1986, they're all too high.
Way over. And of course, we have to notice that this is a logarithmic graph.
Yes.
We're going from 10 to 100 to 1,000 to 10,000. So
yeah.
even those little green lines of SV40 there that are over the mark are like way over if that was on a linear graph.
Exactly.
And if we saw this on a linear graph, all these spikes would be like the Empire State Building, wouldn't they? They'd be sticking way up in the air.
Yeah.
It was on a linear scale. Yep. Some people might think the public is not adequately protected by this.
Nope. And so this is the Irish vials. I looked at, and it's got on the left the Irish and the Canadian vials in linear, and on the right in logarithmic, as most scientists would look at the data, and you can see that on the left, which is in the nanograms per dose, and on the right is in picograms per dose. Linear shows how extremely high these are.
But we have to stress that this is the same data on both graphs, isn't it?
Yes.
Yeah, it's just that the one on the left is in nanograms per dose and is linear, and the one on the right is in picograms per dose and is log.
Yep.
But they're showing the same data.
And then this is the same as the one on the last on the right, and it includes the two Australian files.
Yeah, and again, because this is a log scale, if this was in linear, these lines above the dotted, even the higher dotted line would be like going way up into the air, wouldn't they?
Yep. And this time I did put on 1986 10 picograms per dose.
Yep, indeed. You get a tick for that.
Okay.
Excellent. This is great stuff. What else do you want to focus on, David?
A few key initiatives going on. The Australians, based on this work, launched the David Declaration end of Feb, and it's a return to evidence-based medical science and calls for a moratorium on mRNA technology, as well as recognition and support of the vaccine injured.
Absolutely, yeah.
And
those graphs are really powerful. It's your data straight off the bench, as it were. So this is
exactly.
This is basically a primary source, but that makes this video a primary source publication, essentially.
Yep. And then on the next slide, Europe has launched this North group push to send this data to 22 nations so far, including the UK and Canada. Done. It's based on the whole DNA contamination and the SV40.
Yeah.
And if you go down one more slope to last one, and in Canada this week we've launched our own letter and it is open for signatures at call2halt19.ca.
Yeah, and we can put the links for that, of course. Yep.
Yeah. Okay.
Excellent, David. Thank you so much for giving us this data hot off the press. Really, when can we expect this to be in a publication?
Well, considering my Canadian data has been rejected 4 times and has yet to be sent out for peer review,
But you've been rejected by 4 top journals.
4 top journals so far. Editors won't even send our data out for peer review, and we keep on trying to get this into a publication.
Well, if it's any very minor consolation, more people will probably watch this video than would read some publication anyway. But
exactly. Yep.
But it's still important. It's still important to get it published, of course, for the scientific credibility.
It is. Yeah.
Great. Thank you so much, David. I've got a couple of other questions just for fun.
Sure.
How many types? So viruses can infect eukaryotic cells. They can affect bacterial cells, bacteriophages. They can infect fungal cells. How many different types of viruses are there on the surface of this planet? Just answer to within about 20 or 30.
How many?
It must be like 10 to the 22 or something like that, is it?
Yeah, there's thousands out there. Every bacteria has its own human viruses.
Yeah, crazy. Yeah. I've heard something like 10 to the 22 types. Does that sound?
Yeah.
I mean, that's one with 22 noughts on the end.
Yeah. there's
and then just an easy question to finish. Where did they come from? Now let me ask that question seriously. Is there a recognised set of theories and a recognised literature that is scientifically investigating the origin of 10 to the 22 different forms of virus? Are there reasonable theories that can be debated?
No, I don't think so.
You know, people talk a lot about the origins of life and the last universal common ancestor. That gets quite a lot of attention.
Yeah.
But something as patently obvious as there's 10 to the 22 or so viruses on the surface of the Earth, and that's just accepted. No one seems to say, well, just a minute, where did they come from?
Exactly.
Because they can't have evolved without cells because they need cells.
Yeah.
And they can't all be devolved cells because there's 10 to the 22 of them. It really is a complete ecosystem that just seems to have been planted, and no scientific analysis of why it is like that. It just seems not only a dark hole in biology and virology. It just seems that the hole is so dark that no one even seems to notice that the hole is there.
Exactly. Yeah, I think people have basically asked, but it's things like that you'll never know the full answer.
Yeah.
I
think it's like a complete ecosystem, isn't it? Because we can't survive without the viruses. They can't survive without us.
Yeah.
And all the massive proliferation of fungal cells and bacterial cells and complex cells like we have all have their specific viruses that can infect them, and that's necessary, we would assume, for the normal functioning of the ecosystem. So you've got real chicken and egg there. Which came first, the viruses or the bacterium? And because you can't have one without the other.
Yeah.
Yep. Sorry about those, but they were good fun.
It's
Do you mind
okay.
if I put those? Do you mind if I put those in? They are quite fun.
Sure.
Okay.
It's just so much to think on, and most of these things aren't asked.
Yeah. It just seems curiosity deficit disorder amongst the world's virologists.
Yeah. Exactly.
Anyway, you're doing great work as a virologist. We appreciate it.
All right, John.
The work you're doing is appreciated by millions around the world, even if these miserable scientific journals don't want to publish it. We do. So thank you very much, David. Really appreciate it.
Awesome. All right.
Yeah, thank you.
Take
Talk
care.
again soon.